This is a picture of my father. My father's also a biology instructor. He works at a community college in Oregon. He's been teaching there since 1967 or 68. He's retired about a decade ago or so, but he still teaches there. And he loves to teach, so the apple didn't fall too far from the tree in this case. This is him at the front of the line on a nature walk that he gives to people in his congregation, to his church, to his students, to anybody who wants to go hiking with him. And so this is my upbringing. When I was a young kid in Oregon, weekends, as the son of a biologist, were spent in the car driving somewhere to go on a hike, to go boating, to drive from western Oregon, where I was born, to eastern Oregon, the high desert, where there's basically nothing except for you might go to Malheur Wildlife Station, where there was a takeover by the armed militants a couple of years ago, you might remember. Last year, I think. So driving along the highways of Oregon, up in the Cascade Mountains, in the middle of the summer, it's beautiful, it's green there, not this summer, but it used to be. And my sisters, my two older sisters, my mom, or especially my dad, would say, "Oh, look at all those red columbine" as we drive past, or "Look at those red berries on the side of the road." Wow. I'd say, "What?" And by the way, this was, the example of my upbringing as a child of a biologist. Sitting next to stuffed sage grouse, maybe, or holding a praying mantis, or in the cranberry bogs on the coast of Oregon with my notepad, being a dutiful scientist. So that was my upbringing. I am a mutant. Do you see anything in this picture? Yeah. Okay, if you see something, raise your hand. Besides dots. Okay, what do you see? 3. Okay, does anybody not see the number 3? Gentlemen. 1, 2, 3. Okay, so most of the guys, red-green colorblindness. I can't see anything in that picture. Let me show you what it looks like to me. To me, these two pictures are indistinguishable. What I see in the picture on the left is what you're seeing on the right. No difference to me whatsoever. Look exactly the same. So when my sisters were saying, "Oh, look at all those columbine," monkey flowers if they were red, which they're usually not, they're usually yellow, but if they were red, I don't know what you're talking about. I see a bunch of brown going past. So I'm a mutant. I have one mutation on one of my chromosomes that causes me to be unable to really easily distinguish reds and browns and greens. And this is pretty common. There were a few hands, especially for those of you in the front, that couldn't see. There were a few hands that popped up around the room, people that couldn't see a difference, couldn't see that number. So take-home messages, in some way, we're all mutants, unfortunately. Sorry, I apologize. Ollie brought you here and they give you food and a grocery bag, and then I insulted you. But it's true, we're all genetically different, we're all mutants. And that's one of the first things we're going to talk about after we talk about what is a mutation, what does it mean when we talk about a mutant or a mutation, will be who is a mutant. You already know the answer to that. And by the end of the talk, I'd love it if you will be an ambassador. I'll let you make your own decision. Are mutations bad? Or are mutations bad in this case? So I'd love for you to be able to go talk to friends and family afterwards after I've gone through a few examples of mutations that are pretty common in daily life, and then have you decide for yourselves. Because when I ask my students, I'm teaching mutations this week in genetics class. Feel free to stop by if you want. And at the start of every class where I start talking about genetics, I ask them to free associate. What do you first think of? When I say the word mutation, what's the first thing that pops into your head? And they always say something like, I don't know, what do you think? Which one? Little Green Men. Yeah, it actually turns out Teenage Mutant Ninja Turtles, which is the Little Green Men of this generation. X-Men. So popular characters from comic books and movies. And so mutations are cast usually in a bad light, but they're not all bad. There's the punchline. So the first thing we're going to talk about is what is a mutation, briefly. go into days worth, but we don't have the time. So all I want you to know about mutations is that they're changes to DNA sequences. All of us have DNA, deoxyribonucleic acid, in all of our cells. This is the information that encodes the ability for our cells to divide and their daughter cells to divide to create us, living organisms. So DNA is simply a string of letters, essentially, 4 different letters, C's, G's, A's, and T's. Our DNA alphabet just has 4 letters, not the 26 we have in English. And normally DNA forms a double helix shape inside of our cells, which is what's shown on the left side here. But if we pulled that double helix apart so that we were just looking at the 2 side rails of the ladder side by side, and we actually looked, there would be A's, T's, G's, and C's. And those 4 letters stand for different chemicals that I'm not going to bore you with the names of, but that's what our DNA is. It's a series, it's an individual sequence, just like we speak, we form words and sentences, and the meaning behind those words and sentences have to do with the order of the letters. DNA is exactly the same way. It's basically a cookbook. It's a great analogy that I think almost every biology instructor uses for DNA. Think about your chromosomes, your DNA, as the recipe book for life. Every one of your cells has to have that recipe book. And what's encoded in that book, the chromosomes of the DNA, are things like proteins that actually go about helping your cells survive the day and divide. So, take-home point. We have 4 letters that make up our DNA. So I'm going to use a series of icons to represent the different topics we're talking about. So a mutation then is a change of X to Y in a double helix in the DNA. So X being 1 letter changes into Y, a different letter. For example, on the previous screen here, there was a T next to an A. This is a mutation. There was an A, now it's changed to a G. Simple as that. A mutation. Just a single letter change in our DNA code. More specifically, that could be, as I suggested, an A to a G. So what was an A becomes a G, or becomes a C, or becomes a T. Any one of the other 3 letters. Here's an example of a bunch of different types of mutations that could occur if my name was DNA. So if Joe Ross was in my genetic code, those letters, that J could become a T, and then I would become Toe Ross. So that would be a mutation that does what? What's the effect of the mutation? It changes me into a body part. So mutations can change traits or appearances. What about Joe Ross to Joe Rose? Now I'm a flower. I've changed species. I'm no longer a human. I'm a flower. Or you could think about it another way. Maybe that's a beneficial mutation. Maybe that's a good mutation. I've gone from Ross to Rose. I smell like a flower. It's beneficial. It's a good mutation, maybe. What about deletion of that 1 letter? The E that's the end of my abbreviated or my shortened first name. I go from Joe Ross to Joe Ross. How has that affected me? Now I've had a sex change mutation. I've gone from a male to a female, maybe. Joe Ross to Joe Rots. Bad mutation. Joe Ross to Joel Ross. You add a letter to the DNA code. I call that a neutral mutation. It's not good, it's not bad, it changes me, but not in really a meaningful way. And then we have the addition of an S in between the O and the E. Now maybe I become Jose Ross, and one of the things that I'll mention briefly today is that DNA sequences and mutations have something to do, although maybe not the way you'd expect, with ancestry. So, to finish up what a mutation is and its effect, when you have a single base pair mutation like that A to a G, just a single letter change, a mutation, that can do something as severe as turn somebody able to see red, green, and blue into somebody who sees, I don't know, what are these colors? They're reds or reddish browns. I'm asking you because I don't know. I tried to make those browns, but maybe they're not browns. It's hard to agree on color names. Maybe it's burnt sienna. That's the point. A single change in my DNA sequence, one letter change, takes me from the left side, which is what most of you are able to do, see and distinguish reds and greens, to the right side. So mutations can have big effects, they could have small effects. Joe Rose to Joe Rots. So that's the what of mutations, what mutations are. They can be very small, minute changes in our DNA sequences. Now we get to the who is the mutant, up here at the top. If you look inside any one of our cells, well, guys, because this happens to be the chromosomes of a gentleman, you'll see 23 pairs of chromosomes. So each one of these is a sequence of DNA. Looking under a microscope at human chromosomes, this is what they look like. Our total cookbook, the total length of all of those chromosomes, if you look at the letter sequence, there are 3 billion letters in those 23 pairs of chromosomes. 3 billion, it's a huge cookbook. It encodes around 30,000 different components, or chapters, maybe you want to say, that are essential, most of them, for us to be alive. Now, each of us have 2, or most of us at least, as far as I know, there might be some mutants among you that have more or less. Most of us have 2 pairs of every chromosome. We have 2 of chromosome 1, as you see up here, 2 of chromosome 2, 2 of chromosome 3. Where did we get those chromosomes from is critical to understanding mutation. We got them at fertilization from our parents, yes, absolutely. So at fertilization, at the moment of conception, the sperm from the dad contributes one set, 1 chromosome 1, 1 chromosome 2, 1 chromosome 3, and so forth, from the dad. The other half, the other 23 chromosomes, an equal number normally, come from our mom. Now this is important in understanding who mutants are, because there was a scientific publication study about a decade ago, 2007 down here. Levy et al. Published this in a journal. What they did was, for the first time ever, they took a single genome, all of the sets of chromosomes from one individual, and they obtained the entire sequence, the 3 billion nucleotides of all of the chromosomes. So what they're doing, because half of those chromosomes came from the dad and half came from the mom, they were actually looking at this DNA sequences of the mom and the dad of this individual that they were studying. And they found that in those 3 billion letters, all of the chromosomes, all put together, the whole sequence, there were over 12 million different changes that distinguished the mom from the dad of this individual. 12 million, just between the parents, 12 million differences. So yeah, the parents were genetically distinct, absolutely, totally different DNA sequences. But that's a lot. So something like 0.4%. Any 2 of us, if you compared our DNA sequences, we'd be about 0.4% different if you looked at our DNA sequences, the letters of the DNA. Now, contrast that with the difference between us and our closest living relatives that are a different species, chimpanzees. Some of you probably remember this. I was not born quite yet in 1975. But in 1975, there was a groundbreaking study where Mary Claire King and her advisor, Alan Wilson, who I believe were here in California, I think they were at Berkeley at the time, looked at and compared chimpanzee proteins, essentially DNA sequence, with human to find out how different are we. And this is what they found. So it's not the 0.4% difference between any pair of us, you and I, members of the same species. But we were pretty close in DNA sequence even to chimpanzees, 99+% the same, which means if you look at 100 letters of our DNA sequence and 100 letters of chimpanzee sequence, there'd be one difference. So the point being, not to freak anybody out about how closely related we are to chimpanzees, but to point out that we are all mutants. We're all genetically distinct. We all have sequence differences, every one of us. But at least, maybe the good news, depending on how you feel about evolution, the good news is that we are very, very, very different genetically than chimpanzees, relatively speaking. Yeah. Now, there are important things that happen when you have genetic differences between two species. Is anybody familiar with what happens when you mate horses and donkeys? You make the mule, and the mule is sterile. Fantastic. I thought I was talking to the right audience. That's one of the things that happens when you have organisms that are too genetically distinct. They have too many of those mutations or sequence differences between them. And this is actually how biologists tell species apart. This is one way biologists do this. You mate them. And if they can't have offspring, or if the offspring are dead or sterile, then you say the parents were two different species. Horses and donkeys are two different species. In this case, we know why. In many cases, we don't know why offspring might be sterile or not develop at all. But in the case of horses and donkeys, there's a difference in the number of chromosomes they have. And that's the essential genetic difference. You can consider it a mutation if you want to. The horses and donkeys are genetically different, and so they can't have kids. Or they can, but the kids themselves can't have kids. So that's an important point about mutations. There's a certain point at which we're all genetically different and we're fine, but then if you travel too far down the road of mutation, you're now talking about different species. And you're talking about having offspring that are going to have some problems when you mate them together. And at the end of our time here together, I'm going to share with you one story from my research lab, research that my students are doing that gets at this question of how do you find the mutations or the genetic differences that help you distinguish two different species. But before we get there, I want to make sure I keep the attention on you and what might be relevant in terms of mutation to current topics, society, life. Turns out, you may have remembered this story from a few years ago. NASA has a set of identical twins that happen to be astronauts, the Kellys. And they did a really great scientific study that's only just starting to be analyzed right now. They've collected all the data. What they did was one of the twins was here on Earth, the other twin was at the International Space Station. They wanted to know if there was an effect of space flight on genes, DNA, mutation rates, and so forth. So they took the DNA from Mark and Scott Kelly before whichever one of them it was went up on the space station, Scott. So Scott, they took his DNA before he left and after he came back, and they did the same to Mark. So they've sequenced their DNA. I'm not sure that they've analyzed it thoroughly yet, but they will figure out whether or not there's a difference made by traveling to space and being there for a year. What do you think they're going to find when they look at the before they left for space comparison, the two twins' DNA sequences? They should be the same, I'm hearing, except that even identical twins are genetically different. And identical twins, not just fraternal twins, but even identical twins are genetically different. And that's for a reason that I won't get into now, but if you're interested, I'll tell you afterwards, maybe, to save time. But even twins are genetically different. You can tell them apart if you look at their DNA sequences. So everybody, even if you're an identical twin, even if your twin is not here, you're still a little bit a mutant. Now we're going to talk about context, or where and how mutations occur, why that's relevant. Last year I was approached by Graciela Moreno from our local ABC affiliate. She was doing a story on genetic testing and ancestry, and she wanted my perspective on whether or not this was hokum or whether or not genetic tests really work. So I ordered myself the same set of tests that she had ordered. I put them on my table in my office. This one was maybe the nicer of the two. They send you little spatulas, you scrape some cheek cells off the inside of your cheek, stick it in a tube, you send it to the company. What they do is, they do exactly what I just described. They sequence my DNA, which means they look at certain letters that scientists know are responsible for conferring different traits, or in this case, different letters that we know come from certain populations of humans around the world. By the way, the less nice version is you spit in a cup, and you spit and you spit and you spit and you spit, and you have to get like an ounce or two of saliva, and then you send it to the company and they extract your DNA from it. It took a while. This was faster. But there is solid science behind ancestry testing. And what I got back was, from one of these companies, well, both basically sent me the same chart, was this graph. Based on my mutations that I carry with me, that are known to be specific to certain populations of humans, they told me I'm mostly Western European, which I already knew. My grandfather on my mother's side is a genealogist hobby, but we know that I'm Scandinavian and British Isles, and looking at me, you probably wouldn't guess otherwise anyway. So, yes, I was doing this because I wanted to be, Graciela, her main question to me was, are these actually working? Do they predict accurately? So I figured I'd be the control study. I know my ancestry, so I'll test the genetic testing company, find out if they're, telling me what I think. Yeah, it was pretty accurate for me. So mutations that are specific to each of us, some of them tell us where we came from, where our families, where our family trees, where our ancestors came from around the world. So genes and mutations can tell us something about where we came from. Now, there are also effects of mutations that depend on your circumstances. So in answering that question for yourself, are all genetic mutations bad? It's really important to know that context is important. A mutant you that's living here might be okay, but a mutant you that's somewhere else in the world, or in a different situation, a different climate, maybe cleaner air, might be better or worse off. Normally, we all start life, and it's different these days, we start life breastfeeding. Right? Milk is a required component of our diet for the first months, maybe into years of our lives. But then this weird thing happens to all of us. We can all digest that milk sugar, lactose, when we start life. And then sometime during development as we age, normally, we become lactose intolerant. The enzyme, the protein in our body that chews up that milk sugar so that we can use it as an energy source, turns off. Our cells stop making that protein. And then when you eat cheese or milk or whatever, then you get the unfortunate side effects. That's normal. Now, I'm not going to ask who here is lactose intolerant, but I can see that we've got a predominantly Caucasian audience, which normally we're lactose tolerant. Most of us then carry a mutation that keeps that enzyme called lactase. It keeps it in production throughout our entire lives, so we can eat as much cheese and milk as we want. But this is not normal in other cultures or in other societies. So, in other words, lactase persistence, which is what most of us are if you're lactose tolerant, again, I hate to constantly be ragging on you, but we're all big babies, basically. Somehow, there's a mutation that has allowed that lactase gene that typifies infants to stay on into adulthood. So that's if you're lactose tolerant. Now, as I said, lactose tolerance depends on where you are in the world. Not if you moved somewhere now, but where you were born. It has something to do with your ancestry. In this top graph, which you may or may not be able to see, we've got a map of the world, and the colors represent the frequency of lactose tolerance. So hot colors represent parts of the world where there are lots and lots of people that are lactose tolerant. They can process, digest milk. Notice any interesting patterns? We've got Britain, Eastern Europe, Scandinavia, and where else? Western Africa and the Middle East. And scientists then looked not only at, that's a trait, that's what you can go around and ask people, can you digest milk or not? So that's a trait. The map below it, down here, is asking the same question but about the mutation, the DNA sequence change. So again, red colors indicate parts of the world where there's a particular mutation that allows lactose persistence. So people in Britain, in Scandinavia, for example, all have basically shared the same one-letter change. That's evidence of our relationship, of our relatedness. The peoples of the British Isles and Scandinavia, they're geographically close, they share recent ancestors, they share that one mutation. But what's weird, and what was interesting to these scientists in 2011 who published this review paper, was that that mutation is not present here in Africa or here in the Middle East, where there are people at high frequency that can digest milk. So what happened? It's a different mutation in Africa and the Middle East than the mutation that they were tracking in this map. So one mutation happened in the ancestors of people of the British Isles and Scandinavia. A second, totally different, totally independent, single letter change, or maybe more than one change, happened in West Africa. And probably yet a third one enabled people in the Middle East that are lactose tolerant to continue digesting milk into adulthood. So different mutations can have the same effect. It's not like there's one magic mutation that can cause the same trait change. You can have lots of different types of DNA sequence changes that all result in the same effect, like the ability to digest milk. So where you are in the world has something to do, maybe, with the effects of mutations. So if you're lactose intolerant, now you know you've got ancestry to thank for it, probably. So that's the top half, the who, what, and where of mutations. Where we're heading ultimately is the why, and that's the part where we're really going to get into application. We're going to talk about GMOs, genetically modified organisms. And hopefully that's most relevant to most of you, because we live in a great place that has a lot of fantastic agriculture. Like you heard at the beginning, I love to cook. I love being here. I hope you can tell. But I love the agriculture, and I want to especially make sure that everybody can make good decisions about food products that they purchase. But in between there and now, we're going to talk about how when mutations occur matters, and how mutations actually occur in the first place. I probably should have started with that how, right? I talked about what a mutation was, single letter change, and you were probably have been sitting there for 20 minutes thinking, how does that happen? Now I'm talking about the effects and lactose tolerance and so forth. This is extremely relevant. Hurricane Harvey, if you've been watching some of the news, you've probably seen the reports of the epoxide plant in, I don't remember which town it was, it's near Waco, I think. Was it? Anyway, plants have been exploding. They couldn't keep their chemicals cool because they had power outages and their backup power went out, and their backup of the backup power went out, and finally these chemical tanks started exploding. Well, similar things are happening to other chemical plants, including ones that contain benzene. So the chemical benzene is extremely hazardous, and one just yesterday was reported there was a plume or detection of benzene in the Houston area. And the reason I point this out is if you look at the dangers of benzene according to its safety data sheet, which anybody can get on the internet if you want, just Google or search engine, type in the name of a chemical, and then safety data sheet, you'll get links to what these chemicals can do. And in this case, I'm going to point out specifically, aside from some of the other nasty effects, germ cell mutagenicity, that is, this chemical can cause mutations, not just anywhere in the body, but specifically in germ cells, that would be sperm and eggs. Why is that important? Because if you have mutations in your germ cells, your sperm or your eggs, that means your kids get them. So this is a specific type of carcinogen. We'll talk about distinctions there in a little bit. So it may cause genetic defects, may cause cancer, and a bunch of other nasty things. So people are worried about this in this Houston area especially right now. This is to point out how mutations occur. There are two different ways to get a mutation, if you classify them broadly. One is represented by the sun. That's your environment. So if you happen to be living in Houston and you get a big whiff of benzene, you might have some mutations occur. If, as was discussed in my introduction, you have fair skin and you don't apply sunscreen and you get a sunburn, that sun and its ultraviolet rays, those ultraviolet rays actually physically damage DNA, and that can cause mutations. That's why I have to wear sunscreen and a broad-brimmed hat every time I go outside, and my parents trained me well. The other way to get mutations is to inherit them, as we've discussed. I'm going to elaborate a little bit more briefly. But ultimately, every one of those DNA sequence changes, the 1 in 100 differences in our sequences between your genome and mine, those all came from somewhere. Those all started as initial mutations, not necessarily in me, but somewhere back in my family tree. They were passed down to me in that nasty colorblindness mutation. I blame my maternal grandfather. Well, I'll get to that in just a minute. So another type of, and not to scare you, but there's been a lot of evidence to suggest that charbroiling things creates some carcinogenic materials. That char, which so many of us love, it gives a little bit of texture, it gives some flavor, brown meat is good, contains a chemical called benzopyrene, and they're chemically reactive and they can bind to and harm our DNA. I would not suggest changing your diet at this point, but there are a lot of environmental chemicals that are bad for our DNA and that can cause mutations. So that was all environment. Benzene, ultraviolet light, benzopyrene that might be present in cooked meats, grilled meats, specifically that char. And then you can inherit mutations. So those environmental agents would cause mutations if you were exposed to them in you for the first time. They'd be new mutations. Then there are the existing mutations, like the colorblindness mutation that my maternal grandfather so lovingly passed down to me. He turned 100 in May. So good for him. I hope I get some of those genes. So we get new mutations, and then we have mutations that can be passed down to us. That's how mutations occur. They all start somewhere by being a new mutation. They can be passed to us after that point. And that gets to a really, really important point about worrying about mutation. I'm actually not so worried about skin cancer. You know why? In fact, I'm not really worried about any sort of cancer. Cancer's tricky. But the good thing about most cancers is they might kill you, but I've already had kids. So I know that if I get skin cancer now, I've reproduced, I've had kids. I passed my DNA on to the next generation, that's a good thing. So skin cancer can't be, this tumor, for example, doesn't get passed on to the next generation. Why? Because my skin has nothing to do with creation of kids. If a mutation, ultraviolet light, if I were outside and it was hitting my skin, and if it caused a mutation, a change in my DNA sequence, I don't care if my skin has a change in the DNA sequence. It's just one cell in my skin. So absolutely. So my skin is not part of reproduction. Reproduction happens separately. And as I mentioned a couple minutes ago, and you helped me with this, the mutation has to happen in sperm or in eggs to make it into the next generation. And mostly, by the time we get any sort of cancer, we've already had kids. And that's the reason why cancer still exists. It's a mutation-based process. That is, a lot of cancers are enabled by specific DNA sequence changes that happen in us. But they don't kill us before we have kids. So those mutations pass on to the next generation before we discover that we have or we're going to develop cancer. It's tricky. That's one of the reasons, as was said, I worked briefly at a cancer research center, but that's one of the reasons that it's hard for nature to cure cancer. It's a disease mostly of the aged, and it's because we reproduce before we get it that our kids then are predisposed to getting it, and their kids and so forth. You pass on the genes before you actually die from the disease. So where mutations happen matter. If it's in somatic tissue, if it's in the body, that might kill you, but it doesn't affect future generations. But if it's in the germline, like that chromosome that my maternal grandfather passed on to me that gave me colorblindness, once it's in the germline, it's present in every cell in my body. Sorry to my daughters, they've got it too, because I gave them that chromosome when they were created, and now they might pass it on to sons if they ever have them. They're 9 and 6 now, so they're not thinking about that yet, I hope. I am saving up for weddings, though. 2 daughters. Okay. Time to play a game. Spot the genetically modified organism. Potato, apple, corn, and rice. Rice, I hear all of them. What about potatoes? So, the second thing I want you to take away from this today is not just an answer to the broad question of are mutations bad, but I want you to know the answer to this. So I'm going to go through each of these in a little bit of detail. Okay, here we have tomatillos, upper right. We have gooseberries. We have eggplant. What do all of these have in common? Pop quiz. They're all in the nightshade family, you're right. So this is deadly nightshade. These are all in the Solanaceae plant family. You're welcome, Dad. I threw out a plant name out there. And now we're talking about GMOs. What else is in the Solanaceae family? Bell peppers, tomatoes, and potatoes. All of these fruits are related to each other in vegetable. The problem with the Solanaceae family is they produce a poison called solanine, and you don't want to eat it. That's why it's called deadly nightshade. Presumably none of you have tried it. No one's fallen asleep yet. That's really good. So all of these plants are predisposed to produce this toxin. Now, I'm going to focus on the potato. That was one of the topical GMOs, or potentially genetically modified organisms. It turns out there are a lot of different, and you probably know this, you've got the baby red potatoes, and you've got the russet potatoes, and you've got the, I don't know, the Yukon Gold. Thank you, I was thinking Alaska, and I couldn't come up with it. Yukon Gold potatoes, some are better for roasting, mashing, baking. Potato breeders select for specific properties in potatoes that make them good frying potatoes, that crisp up nicely when they're fried and turn the perfect color that the consumers want. So they have panels and they cook them to different degrees and they have people tell them which color they like the best. So potatoes are bred specifically for some of these characteristics. And a few decades ago, somebody found the perfect frying potato through conventional breeding. They were just mating different types of potatoes together to see what sort of combinations of traits they could make. And to get the right frying texture and color and so forth, they found this potato called the Lenape potato. And here on the left, in the very left column here, you can't read it, that's fine. There are several different cultivars of potatoes. And once they found this really perfect frying potato, they were going to market it. They were about to get it out to market so that people would, Lay's and Doritos or whoever would start buying these and frying potatoes. And somebody thought, thankfully, somebody thought, we should check to see how much solanine this new breed of potato produces. So we look across there into the average amount of solanine produced by those different potato varieties. And that's, I covered up the 1 a little bit. That's 12.51 produced by the Lenape as opposed to 3, 4, 3, 2, and so forth. This one at the bottom that says 10, that's also a Lenape potato. This strain of potato, this breed of potato produces like 4 to 5 times more of this toxin than all of the other potato breeds they studied. So it was thankful that somebody thought to feed this to rats before they decided to start feeding it to humans. They discovered that the rats were not very happy about that. The point of all of this, these potatoes were made by conventional breeding, combining traits from different types of potatoes by mating them together and seeing what their kids looked like, and selecting for the ones that had supposedly the right combination of traits, except for in this case, fried up nicely, beautiful color, deadly. Right? One trait they didn't want. So all of these are caused by mutations, but in this case, one of those mutations, one of those differences between this strain of potatoes and other strains of potatoes made it more deadly. Apple, briefly. There's a company in Washington State that found a way by genetically engineering the apple to keep it from browning when you cut it. It's really simple, actually. A single mutation can turn off the enzyme, the protein in apples, that when you cut the cell open, it releases this enzyme. The enzyme starts chewing on the apple and turning it brown, basically. And so all they did was they used a single mutation to turn down the amount of the enzyme that the apple produces, so when you cut it, the apple doesn't brown. Okay, so you should be thinking, I'm going to ask you to think for yourselves right now as I move on, is this a good mutation or a bad mutation? Here's another mutation, rice. So we need to get through all of these different plants. Turns out wild rice, like shown up here in the top corner, that wild rice, when you thresh it, a lot of the kernels of the rice break. So nobody wants to buy it. So through, again, conventional breeding, mating different types of rices together, scientists, well, not scientists, growers, were able to identify particular lines or strains of rice that wouldn't break when you threshed the rice. The grains were all intact. You could bag it and sell it, and people liked it. This one has a happy ending. This is not at all like the potato story. To make the point about what is a mutation really come home, here at the top is a specific sequence of part of one of the chromosomes from one variety of rice called a Nippon bear variety of rice. It's a Japanese strain that does not shatter when you thresh it. So those are those A's, T's, C's, and G's I was telling you about at the start. And down here is a sequence of the same part of the same piece of DNA from a different variety of rice that shatters. There's one difference. Can you spot it? It's the one that's circled in red already. The point is that's a mutation. That is what a mutation looks like. It's a single letter change from one strain of rice to another. And now one letter difference in this case doesn't make me colorblind. It makes rice either shatter or not when you thresh it. That one letter change makes the tensile strength of the seed something like double for the non-shattering types compared to the shattering types. One letter change. And in us, that would be 1 out of 3 billion letters. I don't know how big rice's cookbook is. It's probably bigger than that. Most plants have DNA sequences that are way longer than most mammals. Plus, a lot of plants have more than 2 pairs of chromosomes, but that's a different story. Now, I know potatoes are important to a lot of us. I know rice is probably important to some or all of us. How about corn? Has everybody bought their sweet corn from Fresno State yet? I stay away when it's in season because those huge lines, but I've had a few different shopping trips to the Gibson Farm Market to buy it recently. This is, I think, if I remember correctly, my last little vignette about plants. Again, one single letter change. One mutation played a role in our ability as humans to access the kernels of corn to be able to use them as a food source. The ancestor of corn is called teosinte. shown here on the left. And then this was growing in Central America, South Mexico, around 3,000 years ago or so, anthropologists tell us. And that's where corn, what we call maize, in other words, was domesticated. About 3,000 years ago in Central Mexico, growers there, people who were farming, found a variety of teosinte that had slightly opened fruit cups. So the teosinte has this really, really tough shell around the fruit. And you can do everything you want to do, but you can't really break into it to get the fruit out. But eventually, a mutation happened by chance. This is one of those, hey, it just happened. Somebody happened to be out in the field one day and spotted teosinte that had a little difference in its trait. That fruit cup was a little bit open. So they took that plant and they grew it, and they grew more of those plants, and eventually they discovered that they could breed from teosinte to some sort of an intermediate to what we now have as corn or maize. So this trait changed over time by conventional breeding, but it was all based on somebody finding that first mutation that made those really tight fruit cups a little bit open. And from there, selective breeding, traditional crop improvement could occur. So mutations have done good things for us, at least in agriculture. All right, so in our last few minutes, let's fill this out. Method of genetic modification for all of these. Only one of those four is genetically engineered. They're all genetically different. So I would claim that all of these are genetically modified foods. Whether that be by crop improvement, that is traditional breeding programs, that would be those three, and genetic engineering. Right, where scientists up in Washington State actually created that apple to turn off a particular enzyme and make it not brown when you cut it. What's the difference between those two? This is really important to understand. What's the difference between crop improvement and genetic engineering? They're both based on DNA sequence changes. It's just in genetic engineering, the scientists make those mutations on purpose and put them into the organism. How does crop improvement work? It's random changes, which means it's less efficient. Because those people in Central Europe, or Central America rather, had to actually find that 1 in 1,000 or 1 in 10,000 or 1 in a million teosinte plants that had the mutation that they were looking for, needle in a haystack, almost literally, needle in a corn stack. You have to wait around for so long to find the mutation that's doing the thing you want it to do. That's a major difference between being any sort of organism and being an engineered organism. It's efficiency. How much time and resource do you have to put into finding the trait that you want? And I'll let you decide whether or not these are good or bad mutations on your own. I want to make one point, though, about the potato. Solanine production went up 4 to 5 times. Crisps nicely, fries nicely. Is that a good mutation or a bad mutation? The increased amount of solanine. What do you think? So it's bad if you eat it. Okay, so I'm going to write down bad. Who would like to disagree? Somebody please disagree. From what perspective is it good? From the plant's perspective, why do the Solanaceae produce solanine? As to predator avoidance. It's toxic. A lot of plants, or at least a lot of parts of a lot of plants, have toxins in them to protect them from predators, from insects and other things. So one of the most important points, I think, if I'm going to make any points at all that stick for you in your mind, you'll go share with friends and family, is, again, this is context dependent. It depends on which perspective you're looking from, whether or not a mutation is good or bad. It's a great mutation in the context of the potato to produce more solanine. Well, it worked out, we never ate them. Okay, so we've looked at examples of the who, what, where, when, why, and how of mutations. I'm going to leave you with one more story. Maybe two if we've got time. How much time do I have left? Half hour? Perfect. Feel free to start throwing stuff out. Let's see. Shopping bags are soft. Start throwing shopping bags when I've run out of time. Now I want to tell you a little bit about the research that I'm doing with my research students next door in the Science 1 building. We study DNA, we're geneticists. And I'm going to tell you about two different types of chromosomes, really briefly. We have those linear chromosomes that I showed you pictures of earlier, the human chromosomes, 46, 2 pairs of 23. We have linear chromosomes. Those are in our nuclei. You don't have to remember this. There is no test at the end. If you forget anything, forget that. And then we also have some chromosomes in our cells that are circular. Those live inside little parts of our cells called mitochondria. Forget that too. Just wanted you to hear it once so that if you remember it long enough to tell somebody, that's fine. I heard it talk about mutations in mitochondria today. Perfect. So two different types of chromosomes. Linear chromosomes that are just a string of letters from one end to the other, and then there are ones that are shaped in circles. And we studied both of those. Mitochondria, the only thing you need to know about them, the mighty mitochondria, what they do is what I've diagrammed here on the right. They convert what we eat, food energy, sugars and so forth. They're a critical part of the process of turning in what we digest and what gets absorbed by our intestines into energy that our cells can use, chemicals that power our cells. They're an integral part of that process. They form sort of the cogs here that I'm illustrating as grinding up the food and turning it into energy. It's not quite what happens, but it's a close enough analogy. And what's most important to know is that one of those cogs or gears is produced by those linear chromosomes. The other one is produced by that circular chromosome. So these components need to fit well together to conduct this process of converting what we eat into energy our cells can use to keep us alive. That's the critical part right here. Two components have to fit nicely together like cogs. So keep that in mind. These are some of my students. This is research they're working on right now. We work with two different populations mainly, an Indian and a Japanese version of this nematode. You're not certainly going to be tested on the name. If you want to know it's pronounced Caenorhabditis briggsii, don't worry. This is a picture of it. It's actually about 1 millimeter long. So it's definitely not to scale here. Thank goodness. I wouldn't have room for them in my lab. And I've color-coded those two different populations we work with, the Japanese and the Indian, as red and blue. One's from a cooler temperature, one's from a hotter temperature, so I figured that was an easy way to remember which was which. So we've got a temperate worm, we've got a tropical worm, both members of the same species. And these are all of my students at their microscopes looking at the microscopic worms. In each of those worm populations, the Indian and the Japanese, we could consider that they have different mutations that let us distinguish their chromosomes. So I color code red chromosomes as the chromosomes, the DNA being in the red population of worms, and the blue chromosomes from the blue population. And we know that worms that are from India are happy and healthy. We can grow them in the lab, they have lots of kids, they live as long as a normal worm of this species should, about 2 weeks or so. And the same is true for the Japanese worms. They're happy, they're healthy, and the blue and the red worms are equally healthy. No difference that we can tell in how many kids they can have, how long they live, and other important characteristics of life. What we wanted to know was, what happens if we mismatch the chromosomes here in the nucleus with the one that's here in the mitochondria? If we combine a red cog with a blue cog, that is the protein that's produced by the mitochondrial genome and the protein that's produced by the nuclear genome, will they work together just as well as blue with blue or red with red to convert food that the worms eat into energy that they can use to live and survive? That was the research question. Remember, this is all about mutations. Those different colors are different colors because those genomes, those chromosomes, have different DNA sequences. Okay, now, I shouldn't have saved the data slides until the end because by now everybody's wondering, wait, didn't he say he was going to ask us a question about the relevance? Well, we're getting to the relevance. But I'm going to show you a few, very limited handful of data slides, and I need to explain what they look like. So bear with me, please. I apologize. This is called a bar chart. This is a box plot. And this is a representation of data. The only thing you need to know is the thick black line represents an average number of measurements, the average of a series of measurements. So if we took the average of the heights of everybody in the room, it'd be like 5'4". So that would be the average. That's all I need you to know about this. So on the bottom of the graph, I'm going to show you again those colored combinations of chromosomes, and we're going to look at how that affects some sort of trait. In this case, I'm measuring, and my students are measuring, a bunch of different traits to assess the health of these mutant worms that we've created that artificially combine the red chromosomes and the blue chromosomes. So here's one of 3 plots I'm going to show you. And again, we're just comparing averages. So which column, if I number them 1 through 4, and we're looking on this axis here at the number of offspring. Higher is more offspring. Who's got the most kids that they're producing? Column 1, 2, 3, or 4? Column 1, it's got the largest thick bar, the largest average. So on average, the red with red lines with red circle, the Indian strain, Indian population produces on average like 220. It's got a lot of kids. They do. They're also hermaphrodites, which means they make their own kids. One hermaphrodite produces its own sperm and its own eggs, and we can talk about that later if you want. It's fascinating. And then the other normal wild strain, blue nuclear and blue mitochondrial, blue lines and blue circles, has on average about a little less than 200 offspring. But we found that there wasn't much change when we made these weird artificial mutants where we combined the red with the blue or the blue with the red. But there are a number of data points down here which I didn't explain to you. There are occasional individuals that have many, many, many fewer offspring. You notice the range here, this is the average. The box tells you something about the spread of the individual fecundities, numbers of offspring individual worms can have. So this was the first experiment we did, and we thought, oh, that's interesting. Maybe there's something to this. Maybe there is an effect of having combined mutations from 2 different parts of the world into the same organism. So then we looked at fat content, because why not? So is there something about are the worms that are the mutants fatter or skinnier than the parents? General effect, the normal wild-type parents, the Indian and the Japanese, have that sort of fat content, and the mutants that we created had slightly higher than normal amounts of fat in them. We made fat worms. Go us. We succeeded. But the important point again was that this is evidence that a simple change between taking an individual that has the blue DNA sequence on the linear chromosomes, if we take that individual and we'd simply get rid of the blue circular chromosome and give it a red circular chromosome, that's the impact. The worm gets fatter. So there's something going on with the mutations that have occurred in the Indian population and the Japanese population that when you put them together, when you make kids that have combinations of those mutations, what happens? they have fewer kids. They're fatter. And does anybody here drink pomegranate juice? I know the Palm Wonderful Company is a good supporter of Fresno State. I'm not being paid to say this, but it's interesting. What I'm measuring here, and what one of my students in particular who published this work measured here, free radical content. That is the number of free radicals that are in these organisms. That's the, if you watch the Palm Wonderful commercials with the samurais and they come in and they attack the free radicals. Free radicals are chemicals that our cells normally produce that damage DNA, by the way. They're mutagens. And so they're bad. So at the top of the scale, we don't want to have high free radical content. We want to be down here at the bottom. And look at the pattern. The two columns on the left, the Indian and the Japanese strains by themselves, the ones we haven't adulterated by mixing and matching their mutations in weird ways, have relatively low free radical levels. But when we make hybrids, especially this combination, the red linear chromosomes with the blue circular chromosome, free radical production, we need to give these worms pomegranate juice, fight the free radicals. So in all of the ways we've tested these worms, we've discovered that when you have a combination of mutations that does not exist in nature, not red with red, not blue with blue, but red with blue or blue with red, we can measure a lot of different ways that those worms are unhealthy. This is basically the horse and the donkey of the worm world. We discovered that the Indian and the Japanese populations of the species we work with are like the horse and the donkey. They're fine by themselves, they're healthy, they're fit, but if you make hybrids between them, when you make their offspring and start combining an Indian mutation with a Japanese mutation, then something goes wrong in the offspring and they have lower fitness. They have fewer kids, they're fatter, they have higher production of free radicals. And so what, in long term, my lab is working on is trying to figure out what are those mutations that cause those effects like lower brood size, increased fat content, increased free radical production. I saved the best story for last, I hope. Tell me later if you want, if this was the best story or not. Lydia Fairchild was going through a process in Washington State, as it says up here, of applying for public funding, public assistance, welfare. And as part of having her kids on welfare, she had to prove that she was the mother of the kids, if the kids were going to get this public assistance. And so Lydia and her husband got genetically tested, and the daughters got genetically tested. And the DNA evidence came back and said, Lydia, you're not the mother of these kids. Your DNA does not match the DNA of your kids. And of course, she freaked out. And this is, I hate to make this funny, but this is the best quote. I knew that I carried them and I knew that I delivered them. Of course. Yeah. But how do you prove that? To a court, especially. So here's what you'd think. Here's Lydia. Now, because I don't like to use a lot of color in my talks, because I can't see a lot of the colors sometimes, I've used black and white here to represent different DNA sequences or different mutations, different genetic backgrounds. I'm not meaning to indicate that this is African American versus Caucasian or anything like that, just black and white. So if she has black DNA, and if her husband has gray DNA, the two colors being distinguishable by different types of mutations, we discussed that. Two parents have different DNA sequences. So when those two DNA sequences combine in the kid, the daughter in this case, her DNA should, half of her DNA should be black and match the mom, and half of her DNA should be gray, match the dad's DNA sequence. Okay? It's based on biological principles. But instead, this is what they found, basically. So this is a summary of the results. Mom's DNA had the black sequence, dad's DNA had the gray sequence. When they genotyped the daughter, when they sequenced the daughter's genome, genetically tested her, she was half white, half gray. So that makes you think what? So the kid matches the dad, but not the mom. So what happened? What's the punchline of the story? Well, the court would think, yeah, someone else was the mom. A mom with the white DNA sequence, not Lydia, but some other woman, had been the mother of the kid that she was claiming was hers. So that's where the whole thing started. Was the court says, wait a second, your DNA doesn't match these kids, you're trying to get public assistance for them. So I'm here to tell you what happened. It turns out that weird things happen during early development. In this case, here's Lydia's mom. It turned out this happens maybe more frequently than you'd think, too. We talked about having sex. When Lydia's mom had sex with Lydia's dad, or whoever the male sperm contributor was, 2 fertilization events happened at the same time. With different eggs, 2 sperm. So this would normally be fraternal twins, non-identical twins, when you get 2 eggs that were released at the same time and 2 sperm that fertilize them independently. So that happened in Lydia's mom. What happened after the weird part? So as those embryos start developing in the uterus, the circle, they start dividing. So the one black DNA sequence embryo divides from 1 cell to 2 cells, and 2 cells to 4 cells, and so forth. And the same thing happens to this other embryo. What happened in this case was those 2 embryos were close enough together in the uterus that at some point they divided and divided and divided, and they merged together into a single embryo. Cells can do this. Of course, it's really rare to have 2 fertilization events happen at the same time. It's even rarer to have those 2 embryos implant in the uterus close enough to each other that this could happen, but it does occasionally happen. So of course, the point is the developing embryo is the mom in this story, Lydia. She is a genetic chimera. Half of her body comprises cells and DNA that originated from that one fertilization event. Another part of her body originated from the other embryo. So 2 embryos became one individual. So half of her, maybe not half, some portion of her body has one genotype, one set of DNA sequences from mom and dad. The other part of her body has different DNA sequences, both still from her parents, but they're different depending on which part of her body they look at. So what I didn't tell you, but the punchline is that what they did when they did DNA testing was like the DNA test I told you about that I took. They used a cheek swab. So they took DNA from her cheek, they tested her, they said, your DNA from your cheek does not match your daughter. Guess what they should have done? They should have taken DNA from the genitals, basically, where the eggs originated from. So they did a cervical smear of Lydia, and they discovered, oh, in that area of your body, you match your daughter. After years of court battles and trying to figure out what was going on. So that's her, half of her body, probably not exactly half, but half of her body having one sequence of DNA, and another part having a different sequence that's the part that contributed the egg to the fertilization event that led to her having her daughter. And that is why you get headlines like this. Woman was her own twin, and the twin was the mother of her daughter. Now, I'm just 2 minutes or 1 minute from done, so I want to point out an important thing here. It's an interesting story, And it has to do with genetics and development, and a little bit about mutation. The only part about the mutation that's important here is that scientists in the courts were using mutations to do DNA testing. The point, of course, is that DNA testing is not always accurate. Context is important. In this case, the context was, what part of the body are you looking at when you do genetic testing? And I also want to point out that chimerism up here doesn't really have to do with mutation. It does in the sense that it's combinations of two sets of cells that probably have two sets of DNA. It's not in itself mutation-based, but chimerism does happen. There might be some chimeras in the room right now. We don't know because most people don't get genetically tested at different body parts, let alone at one body part. Last point. If you take anything away, maybe the most important thing, again, to realize is to embrace uniqueness. We're all mutants. We're all genetically different, but we're all humans. We're 1 in 1,000 letters different from each other. We're 99.9% identical, and especially given things that have been happening recently, I'd like to advocate for treating each other as such, very similar in all humans and not so much different. Okay. Now, here's my contact information.